Hello everyone, welcome to the class. So today we are going to talk about the human health and disease. We are going to start the new chapter, human health and disease. Before going to complete that, in the last chapter, biomolecules, there are some topic left. We will revise that and then we will go to the next. Okay? Yeah. Okay. So 1 minute. Yeah. Fine. So before going to start the class, obviously there is a question for you. So here is a recall question for you. So the question is, this is the correct answer. Okay. Yeah. Option A is enzymes are highly specific. Option B, enzymes are denatured at the high temperature. And option C, enzymes require optimum pH and temperature for maximum activity. And option D, enzymes are mostly proteins, but they are lipid also. Okay, so you can answer me to this question. I will run the poll. Okay, I will run the poll. You can answer me, or else you can answer me in the chat box also. No problem. Okay, yeah. Can anyone answer me in the chat box? Yes, I'm waiting for your answer. Answer me. I will give you 1 minute time for you. Answer me in the chat box. Okay. So just think which will not suitable. Okay. So just remove the which is the option is not correct. Then you will come to the which option is correct. Okay. So they are telling that which is incorrect. Okay. Select which are the correct answers. Okay. So which are the correct answer you select. Then obviously you will get to know that this is a not correct answer. Okay, student. Just try it once. I need answer from you all. Fine. Yes. So which is the correct answer? Yeah. So I got the correct answer. So that is enzymes are mostly proteins, but there are some are lipids. So that is not a correct answer, right? So we know that the enzymes are usually a proteins, but some are ribozymes. So that is except the ribozymes, all are the protein itself, right? Okay. So let us select the answers. See here. Option D is the correct answers, right? So we will study about the which are the other options. Enzymes are highly specific. Yes, enzymes are highly specific. Enzymes are denatured at high temperature. Yes, they are proteins. They are denatured at the high temperatures. Then coming to the enzymes require optimum pH and temperature for maximum activity. Yes, they require optimum pH and optimum temperature for their maximal activity. Fine. So then wrong option is they are mostly proteins, but they are not lipids, right? They are not lipids. So that's why this option is wrong. So they have told you that incorrect option. So that's why the option is correct answer. Okay. Fine. So we are talking about the inhibition of the enzyme activity. So what is this actually inhibition mean? So see here, reduction or stoppage of 1 minute. So student, so enzyme, what is the function of the enzyme? It is going to convert the substrate into the product, right? So if anything happens to enzyme, which will stop the conversion of substrate into the product, so that we will call it as inhibition. Okay, inhibition may be of any kind. Okay, so it may be from the pH, or it may be from the temperature, or it may be from the any external chemicals. Okay, so that is what we will call it as inhibition. Inhibition is what the enzyme cannot convert the substrate into the product. That we will call it as inhibition. Okay, so let us see here. The reduction or stoppage of activity of enzyme due to the presence of adverse condition or chemicals is called as inhibition. I have told you adverse condition means in a decrease or increase in the pH. We told that our enzyme is very narrow, it will work in narrow pH and temperature. It requires the specific pH and requires a specific temperature, right? So something happens, something increase or decrease in the pH or temperature. So that is adverse condition for the enzyme activity. Okay. So that is the first thing. And any chemical, external chemicals or any substrate. So that is also, sorry, any product that is also acting as the inhibiting chemical. Okay. So then coming to the inhibition, there are two types. Okay. So first one we'll talk is the reversible inhibition. Okay. So what do you mean by reversible actually? What do you mean by reversible? Something it is damaged, but it can be reversed into the normal, right? That is what we will call it as the reversible inhibition. Reversible, right? So same thing happen here. So the reversible inhibition is, see here, it is a type of inhibition. Okay, it is a type of inhibition. So the active site of the enzyme is blocked, okay, by the other chemical substances. Once the chemical substances was removed from the active site, the enzymes become active. Okay. So what it means exactly like, so students remember here, in the reversible inhibition, what will happen? There's some chemicals is going to come and bind to the active site of the enzyme, right? So thereby it is blocking the active site where substrate cannot come and bind to this active site, right? So later what will happen? So by removal of that inhibitor or removal of that chemical, it will reframe the active site again, and the enzyme can work as it is like before. Okay, so that is what we will call it as a reversible inhibition. After removing the inhibitor, our enzyme is acting as usual. It is converting the substrate into the product. Okay, so here the inhibitor not damaging the active site of the enzyme. Okay, so that is what we will call it as a reversible inhibition. Okay, so remember here. So what here it is happening? So inhibitor not damage. Okay, inhibitor not damaged active site. Okay, inhibitor not damaging the active site. It is just blocking the active site, right? It is just blocking active site. Okay, so this is what happened in the reversible inhibition. I think you understood this. Okay, fine. So next we'll come to the irreversible inhibition. So just think about this. What happened in the reversible inhibition? The active site is not damaged, right? Just it is blocking the active site. But what happened in the irreversible inhibition is the inhibitor will come and bind to the active site of the enzyme, but it is damaging the active site. After the removal of this inhibitor from the active site, so that damage is not be removed. The damage is happened to the active site where the substrate cannot come and bind to the active site of the enzyme. It is completely damaged. Okay, so that is what we will call it as the irreversible inhibition. What happened in the irreversible inhibition? See here, irreversible inhibition is a type of the inhibition where active site of the enzyme was permanently damaged. Okay, permanently damaged where it cannot be. Where the substrate cannot be come and bind to the active site again. Okay, so that is what we call it as irreversible. Okay, fine. In the reversible inhibition, there are two types. So that we are talking is the competitive inhibition, and second one is the non-competitive inhibition. What is this competitive inhibition? What is this non-competitive inhibition? Let us start. Okay, let us study about this. Okay, see here. So what happened in the competitive, just think about what is the competition. So here, if for NEET exam you are writing, right? So many students are competing, right? So that we will call the competitive exam, right? Same here, what will happen? So the substrate and the inhibitor, okay? The substrate and inhibitor having the same structure. So they'll compete for the active site. So what, the enzyme is only one and the active site is also the one, right? See here, the active site. So this is the active site of the enzyme. Okay, this is the only active site, right? So for this active site, two people will fight. Okay, so which are those two people? First one is the, which are those two people? First one is the substrate and second one is the inhibitor. Okay, so two of them will fight for only the active site. So that's why we call it the competitive inhibition. What will happen in the competitive inhibition? Where substances similar in the structure to the substrate competes with the substrate for the active site. Okay, so see here, the substrate is similar, right? The substrate inhibitor is look similar like the substrate. Okay, so that's why it is competing with the active site. See here what will happen. Whenever this substrate will come and bind, it is converting into the product. Okay, it is converting into the product. The substrate will bind to the active site and it is converting into the product. But what will happen? The inhibitor will bind. This will not convert into the product. Not convert into product. Okay. So then what it will do? It will block the active site. Okay. It will block active site of the enzyme. Okay. It will block the active site of the enzyme. So this is what our competitive inhibitor will do. Okay. So see here, where the substance similar in the structure of the substrate compete with the substrate for the active site, but could not undergo chemical reaction, right? So when the chemical reaction was undergo means it should be converted into the product, right? It is just blocking the active site of the enzyme. Okay. So once it is blocked, the other substrate will not come and will bind to that. Okay. So that is what it is giving. Just think about. So whenever in your school, the bench, where you sit usually, there your friend came and sat means, so here that place was blocked, right? You cannot come and sit there, right? Either you have to tell your friend to get from this place or you have to tell them to, or you have to sit there, right? So you have to sit there means there is no place. It was blocked by your friend, right? So that is what happened in the competitive inhibition. Okay. So fine. So what is the substrate analog? See here what happened. The substance which is similar in the structure to the substrate is called as a substrate analog. I have told you. So here our substrate, here our inhibitor is similar to that of the substrate, right? So thereby it is blocking the thing, blocking the active site, right? So same thing that that type of the inhibitor we will call it as the substrate analog. What is the substrate analog? That inhibitor is similar to the structure of the substrate. That we will call it as the substrate analog. Okay, student? Fine. Here I have taken the example. So I think you have studied the succinate dehydrogenase in the TCA cycle, right? TCA cycle or Krebs cycle, you can tell, right? TCA cycle. See here. So here, succinate dehydrogenase enzyme. So here the succinate dehydrogenase is a enzyme. It was inhibited by the malonate. Okay. It was inhibited by the malonate. Which is the inhibitor here? Malonate. Inhibitor is the malonate where it looks similar like the succinate. Okay. It looks similar like the succinate. Here succinate is a substrate, right? Succinate is the substrate. In the place of succinate, this malonate will come and bind and it will blocking the enzyme active site. Okay. So this is what happened in the competitive inhibition. So same thing. What happened in the competitive inhibition is, so let us talk about the Km and Vmax. Okay, student. So Km is going to be increases. Okay, Km is going to be increases and Vmax is going to be constant. Okay. So what happened when the Km was increases? What happened when the Km was increases? You know, Km is the Michaelis-Menten constant, right? So I have told you that whenever the enzyme is having the high Km value, it is having the less affinity for the substrate, right? It is having less affinity for substrate. Okay. So this is what happened in the competitive inhibition. Okay. So I think you understood this. If you have any question, you can ask me. So fine, let us go to the next. Allosteric modulator. So student, this allosteric modulator, we also call it as allosteric modulators, we also call it as the non-competitive inhibitors. Non-competitive inhibition. What is this non-competitive inhibition? Just see here. So here the substrate and the inhibitor does not look similar. Okay, does not look similar, and both of them will not compete for the active site. Okay, so here the substrate will bind to the different site and the inhibitor will bind to the different site. Okay, so that's why we'll call it as a non-competitive inhibition. But there also it is affecting, right? There also it is affecting. How it is affecting? See here, the substance which is bind to the enzyme other than the active site. So your inhibitor not binding to the active site, but it is binding to the allosteric site. Thereby, see here, before it is binding to the allosteric site, the enzyme was active. The enzyme was active and its active site was active site structure was not changed. Okay. So, but whenever this inhibitor come and bind to the bind to the allosteric site, okay. See here, there are two sites in the enzyme. Okay, 1 minute, I will tell you. Two sites. One is the active site. This V shaped is there, right? So that is the active site. And so this shape is there, right? So this is what we will call it actually allosteric site. Allosteric site. Okay. So what happened here? See here. The substrate is coming and binding to this site, then it is converting into the product. Okay. So whenever this inhibitor, see here, this blue color inhibitor bind to this allosteric site, this active site of the enzyme is going to be damaged. Okay. It is going to be changed. On that time, it is not going to be bind to the active site of the enzyme. Thereby, it is damaging. Thereby, it is damaging the active site. The enzyme cannot cannot bind to the substrate and convert the substrate into the product. Okay. So this is what happened in the allosteric modulators. For example, if you take the example of the glycolysis. Okay. So in the glycolysis, the allosteric modulator is the ATP. Okay. Allosteric modulator is the ATP. So already ATP is synthesized, right? So what happened? The enzyme is pyruvate kinase. Pyruvate kinase enzyme. Okay, student, pyruvate kinase. When the pyruvate kinase was binded by the ATP, it is inhibited. Okay, inhibited. You know what is the ATP, student? Can anyone tell me what is the ATP? ATP is the end product of the glycolysis, right? End product, right? End product, right? Glycolysis end product is the ATP and the pyruvate, right? So when the pyruvate kinase was inhibited by the ATP, that we will call it as the allosteric modulators or the non-competitive inhibition. Okay, let us see here. 1 minute. Okay, so see here. It is other than the active site, modify the enzyme so that the substrate cannot bind to the enzyme and catalyze the reaction. Okay, so the example is end product of the reaction inhibit the enzyme. So that only I have taken the example. Here enzyme is the pyruvate kinase and the end product is the ATP. So here the end product ATP is going to be inhibit the activity of the enzyme. So another thing here you have to remember that about the Km and Vmax. Can anyone tell me what happened to the Km and the Vmax? Can anyone tell me? Km is constant. Okay, Km is constant in the allosteric modulator because there is no competition, right? So if there is no competition, means how come the Km is going to be decrease or increase? It will be constant. Okay. So then coming to the Vmax, Vmax is going to be decreases. Enzyme maximum activity is going to be decreases. Okay, student. This is about the allosteric modulators. So do you have any questions, Anshika Tiwari? Anshika Tiwari, do you have any questions, ma? Then coming to the cofactors. What is a cofactor actually? So student, you know that. So every enzyme is made up of the protein, right? It is the enzyme is made up of the protein. If the enzyme only made up of the protein, that we will call it as a simple enzyme, right? Simple enzyme. So if it is made up of the other than the protein atom, other than the protein, we will call it as a conjugated enzyme, right? Conjugated enzymes, right? See here. So here, for example, if you talk about the conjugated enzyme, holo enzyme. What do you mean by holo enzyme, students? Complete enzyme, we will call it as a holo enzyme, right? This holo enzyme has a two part, okay? One is the apo enzyme part, apo enzyme, and another we will call it as the cofactor. So in the holo enzyme, it is containing the apo enzyme plus cofactor. That we will call it as the holo enzyme. Okay. Here, the cofactor means inorganic factors. Okay. Inorganic factors. What are the inorganic factors, student? For example, if you take the enzymes like if it is having the Mn2 plus, Mg2 plus, right? So these all are the inorganic components, right? So apo enzyme protein part plus Mn plus 2 or Mg2 plus, that we will call it as the conjugated enzyme. If you take the example of the molybdenum, if you take the example of the nitrogen reductase, right? So what is the cofactor it is having? Molybdenum, right? So that type of the enzyme we will call it as the conjugated enzyme. Okay. So that cofactor is the inorganic part and the protein part of the enzyme is apo enzyme. Okay, student. Fine. So another thing is there in the holo enzyme. Okay, if the holo enzyme it is made up of apoenzyme, okay? Apoenzyme plus coenzyme. Coenzyme, okay? Student, coenzyme means organic part, okay? Coenzyme means organic part, we'll call it as a coenzyme. In the coenzyme, you can take almost, it will be made up of vitamins only, okay? It is made up of vitamins only. You can see here example NAD, NADH, FADH and the vitamins, okay? So let us go to the, for example, if you take the succinate dehydrogenase, succinate dehydrogenase require the NAD plus, FAD plus like that, right? Dehydrogenase enzyme, right? So that part they require is the organic cofactors. Okay, fine. So let us go to the another type of the enzyme is there. So that will act as the prosthetic group. Okay, prosthetic group, prosthetic group. So student here you have to remember that whereas the cofactor and coenzymes are loosely arranged, okay, loosely attached to the protein part, okay. So whereas prosthetic group is tightly attached, okay, so that are tightly attached to the enzyme, tightly attached to protein part, okay. So that we'll call it as a prosthetic group. Example, if you take heme group, heme group in the catalase enzyme, okay. So this is about the prosthetic group. So do you have any confusion here? Holoenzyme is made up of the apoenzyme, so that is the protein part, and the cofactor, if the cofactor means that is the inorganic factor, and if it is the organic part means, so that is the coenzyme, okay? So that are loosely attached whenever we come to the prosthetic group, so they are tightly attached. Okay, student? Fine. Do you have any questions here? Okay. So if you don't have the question here means I have a question for you. Answer to this question. Anshika, you can answer to this question, which is the correct answer. Can you try? Students, I want you to try this question. This is very easy question. We have learned in the structure of the protein. Tertiary or quaternary structure of the protein. Can anyone answer me? Which is the correct answer? Yes. So the correct answer is tetramer of the two different subunits, right? So here what we are asking, the quaternary structure of the human hemoglobin is best described as, okay? So think here. So the hemoglobin is made up of 2 alpha and 2 beta, right? So think here, then how many this thing is there? Tetramer, right? So it is having the 4 subunits. So that's why I'm calling as tetramer, right? How many different subunit is having? It is having the four different subunit? No, only alpha and beta is there. So that is the 2 different subunit, right? So that's why tetramer of the 2 different subunit is the correct answer. Okay, 2 different subunits. Okay, student, fine. So this is how we completed our, this is the last slide. We have completed our biomolecules chapter. So do you have any questions about the biomolecules? Kindly ask me. Do you have any questions, student? Fine. So we'll go to the human health and disease chapter. Okay. So fine. So let us start the class. So what are the things we are going to study in this? Okay. So student, we first the content we'll study, then we'll go to the next. Okay. So first we are going to study about the common diseases in the humans. Which are the common diseases? For example, bacteria, virus, protozoa, helminths disease. So this is all the thing we are going to study. Okay. So the second thing, immunity. Immunity we are going to study. Okay. So the last thing was AIDS and cancer. And last thing was the drugs and alcohol abuse. So these all the things we are going to complete. Okay. So then here is a question for you. Can anyone answer me which is the correct answer? Which one of the following tests is considered as gold standard test for the diagnosis of COVID-19? Can anyone tell me which is the correct answer? That is real-time RT-PCR, right? Yes. So let us go to the next. Okay. So what do you mean by actually health is? Can anyone tell me what does it mean by actually health is? We can't tell that if the person is not having any disease, means we can't tell that he is healthy, right? Yes. So if we are telling any person is a healthy person, means he must be physically fit, mentally fit, socially well-being, and the emotionally fit. Okay. So that's why we'll call it as a healthy person. So that is a well-being state. Okay. So when it comes to the disease, what is the disease? So when we are not having the disease, our body function is correctly, right? Our body functioning correctly. So whenever disease we are having, so there is an alteration of the body function is going to be happen, right? So that is what we will call it as the harmful deviation. Okay. So see here, what is the disease? Disease is nothing but the harmful deviation. Okay. So the malfunctioning of the body with the specific symptoms. What happened? So if you have a fever means what is the symptom? You can see. So you are having the body temperature, high body temperature, right? Yes. So malfunctioning of the body, you can't work the correctly, you can't work correctly when you are having the fever, right? Malfunctioning of the body with the specific symptoms it is showing, right? So that is what we will call it as a disease. For particular disease, we have the particular symptoms, right? Yes. So let us go to the next. Fine. Whenever we are talking about the diseases, there are two types. Okay. First one we will call it as a congenital disease, and second one is the acquired disease. Okay. So do you know what is the congenital disease, student? Nothing. Congenital means the disease which is we are having from the birth, we will call it as a congenital disease. For example, you can take the any chromosomal disorder, like you are having the Klinefelter syndrome, Turner syndrome. So these all the things will come from the by birth, right? So that's why we'll call it as a congenital disease. Okay. So some diseases will acquire in our lifetime. Okay. So for example, if you take any viral disease or bacterial disease, so these things we will call it as the acquired disease. Okay. So in the acquired disease, there are two types. Okay. So one is a communicable disease and another one is a non-communicable disease. What do you mean by communicable disease? It will be communicable. Okay. From one person to another person, it is transferring. That we will call it as communicable disease. See here, in the communicable disease, there are two types. One is a contiguous and another one is a non-contiguous. Contiguous means what? The contiguous means it is going to be spread through the direct contact. That type of disease we will call it as a contiguous disease. Okay. So that is going to be transfer direct contact with the direct contact. Right? Then coming to the non-contiguous, it is the indirect. Okay. That is the indirect. For example, through the any transfer, for example, through the you're getting the malaria through the female anopheles mosquito, right? Intermittent. So that we will call it as a non-contiguous. Okay. So then coming to the non-communicable diseases, these are not going to be communicable through the talking with that person or sitting with that person or sharing the things with the person. Okay. So that is what we will call it as a non-communicable disease. For example, if you take deficiency disease, if some person is having the deficiency, for example, protein deficiency, marasmus and kwashiorkor is going to be common, right? So that is a protein deficiency is not going to be spread to the another person, right? That is a non-communicable. The neurodegenerative disease and addiction and allergic reaction and mental disorders and the cancer, right? Cancer is a non-communicable disease. Okay, it is a non-infectious disease. It is not going to be transferred to the another person, right? So these all the things will come under the non-communicable diseases. Okay. So then coming to the two types of the how we are going to get the disease. Okay, one is through the exogenous and another is through the endogenous. What do you mean by exogenous? From the outside the body, outside the body. Okay, student, we are getting the disease from the outside the agent, outside the body agent that we will call it the exogenous. Okay, so then coming to the endogenous, the disease causing agent here is from the inside the body itself. Okay, for example, if you take the diabetes, okay, so which is the disease causing agent here? So glucose, right? So here the glucose concentration was increases in the blood. So this has become the chemical agent which is causing the disease, right? For example, if you take the uric acid, right? So that is the nitrogen metabolism last product, end product is the uric acid, right? So because of the uric acid increases, you are going to get the arthritis disease, right? Arthritis. So that is what we will call it, the endogenous. Here you can take uric acid, uric acid. So these are from inside the body. Okay, these are from inside the body. Okay, endogenous agent. So then coming to the transmission of the disease, yes, I have told you from the cough or sneezing or from the droplet infection, you are going to get the diseases. So that we will call it as the direct transmission. Okay. So the indirect transmission is from the format bond. Format you can take. If the person is holding the pen, or if the person is touching anything, if we touch that again, means we are going to get the disease. That is from the indirect transmission, or from the vectors, you are going to get the indirect transmission, right? So vector is a malarial vector. You can take the female Anopheles mosquito. So some housefly, you are going to get the diseases, right? So these all the thing we will call it as the indirect transmission. Okay, fine. So then coming to the, see here, we are given direct transmission is contact with the infected person, and droplet infection, contact with the soil, and animal bite, and through the placenta. Okay, so you have to remember this all the thing. So these all are from the direct transmission. Okay, without the intermediate. The intermediate is not required here. Okay, see here in the picture, if some person is sneezing or sitting beside, so from that person you are going to get the disease, sneezing or coughing. So from that you are getting the disease. That we will call it as the direct transmission. Okay, so animal bite, contact with the soil, these all the things. So then coming to the contact with the infected person, for example, if you take the smallpox, chickenpox, and the ring-worm disease, so these are all the things you are going to get with the contacting with that infected person. Okay. So then coming to the next, so that is the droplet infection. So tuberculosis, influenza, cold, measles, and whooping cough. So these all the diseases you are going to get through the droplet infection. It may be a sneezing, or it may be a coughing, or it may be a spitting. Okay. So these all are from the droplet infection. Okay. So see here from the soil. So soil you are going to get from the soil you are going to get the tetanus. Okay. So which is the tetanus, student? Can anyone tell me which is the causative agent of the tetanus? Can anyone tell me what is the causative agent of the tetanus? Jainvi, Barsha, Jainvi Kudra, Jainvi Malhar. Can anyone tell me which is the causative agent of tetanus? I need answer from you people. Can anyone tell me? Clostridium tetani, right? Some students are telling, right? Yes. Clostridium tetani. Tetani. Okay, student. Tetanus is caused by the bacteria. That is, we call it as a Clostridium tetani. Okay. So then coming to the rabies. Rabies is a viral disease, right? So it is going to be caused by the animal bite. So that is also through the direct contact. Okay. So here rabies is going to be spread through rhabdovirus. Okay. 1 minute. Rhabdovirus. Okay. Rhabdovirus. Okay. The causative agent of the rabies is rhabdovirus. So that is a viral disease. So then coming to the through the placenta, which are the disease going to be get? See here, German measles, HIV, and the syphilis. If the mother is having the HIV, means the child is also going to be get the virus, right? So the AIDS. So that is the going to transfer through the placenta. Okay. So remember here, student, they may ask you this question, this type of the question. So you have to ready for this type of the questions. Okay. German measles, HIV, and the syphilis are going to be transferred to the placenta. Okay. Then here is a question for you. Which one of the following disease cannot be communicated through the droplet infection? So like sneezing, and I have told you coughing and spitting. This disease is not going to be caused through that. Okay. So I will run the poll. You can answer me in the poll. I'm launching the poll. Answer me in the poll. Or you can answer me in the chat box also. Welcome to the class. Jainvi, Subbalakshmi, Jainvi Kudra, Barsha, and Jainvi Malhar. So answer me in the chat, or answer me in the poll. Which one is? Very easy question. So student, answer me fast. Answer me fast. In the chat box or in the poll. Fast. Fast. No one answered till now. No one answered. I need answer from everyone. Fast. Please be interactive. Okay. So the correct answer is HIV/AIDS, right? So HIV is not going to be transferred through the droplet infection, right? These all the things when it comes to the influenza, measles, tuberculosis, this all the thing is going to be transferred through the droplet infection. Okay, fine. So then coming to the vectors, through it may be the indirect. So I'm telling that it is through the indirect transmission. It may be through arthropod vector or vehicle-borne method, for example, air, water, soil, this thing, and airborne method, format-borne method, and the human carrier. Okay. So when it comes to your format-borne method, I have told you that, for example, knife is used by the infected person. If another person touches the knife or pen or book, means it is going to be transmitted through that to that book or the pen. So that is what we call the format-borne method. And coming to the human carrier, so the famous example for the human carrier is what? Typhoid, right? Yes. Typhoid Mary, right? Yes. So her name is the Mary Malone. So Mary Malone is the professionally she is a cook, right? So from the Mary Malone, so the typhoid was transmitted to the so many people, right? So many people, because she is a carrier of the typhoid, right? So that's why her name is Mary Malone, but they are given the name Typhoid Mary. Okay, Typhoid Mary. Typhoid Mary is the example of human carrier. Okay, so her name is Mary Malone. Mary Malone. Okay, student? Fine. So see here from the arthropod vector, rat flea. From the rat flea, you are going to get the plague, right? And from the housefly, you are going to get the typhoid, cholera, and dysentery, right? And from the mosquito, malaria, dengue, filaria, and coming to the sleeping sickness, it is from the T-safe fly, important. Okay, student. So it is going to be caused from the trypanosoma. Trypanosoma. Okay, trypanosoma, we can call it as a cruzi or gambizi. It is going to cause the sleeping sickness. Okay, trypanosoma, you remember. So that is the genus name. Okay, so then coming to the leishmaniasis, it is going to be spread through the sandfly. Okay, it is an intermediate again. Okay, fine. So how the disease is going to be caused? So there are two ways. Okay, one is through the tissue damage and another through the toxin secretion. Okay, so what is happening in the tissue damage? See here, in the tissue damage here, I have taken the example of rabies. Okay, so student, what happened in the case of the rabies? It is going to cause by the rhabdovirus. Rhabdovirus. Okay, so here you can see here in the picture, the dog is biting the person, right? The biting the person where it has bitten. So here the virus is going to be multiplying the number. Okay, it is increases in the number, multiplication. Okay, so whenever the number was increases or multiplied more, it is going to be transferred through the peripheral nerve. Okay, 1 minute. Peripheral nerve. Okay, it is going to be spread through the peripheral nerves. Okay, from the peripheral nerve, it is reaching to the spinal cord, and there it is going to reach the brain, central nervous system, and it is going to be damage the cerebral cortex. Okay, it is going to be damage the cerebral cortex. Okay, so that's why through the tissue damage. So what it is doing, it is a damaging the tissues while it is moving in the spinal cord, right? So you see here, it is damaging this way, right? Damaging in this way, yellow color part, right? So that is what we'll call it as a tissue damage. Okay. So then after that, what happened? When it is reaches the brain, so then the multiplication will happen. Then that virus is reaching the saliva of that person. Okay, saliva of that person. So student, we will call the rabies as the hydrophobia also. We'll call rabies as the hydrophobia also. Why we will call it as the hydrophobia means the person with the rabies disease, they cannot drink the water. Okay, so cannot drink the water because this is going to damage this trachea. Okay, damage the trachea. Thereby, the person cannot drink the water. So whenever he is having the thirst and wanted to drink the water, there drinking the water, while drinking the water, it is causing the pain. Okay, so that is the reason he cannot drink the water and he is having the fear about the water. Not only about the water, any liquid items. Okay, so that is about the tissue damage. Okay, rabies. So then I'm coming to the exotoxin and the endotoxin. So just think, student, whenever the bacteria was entered our body, so it is going to release some type of the chemicals. Okay, some type of the chemicals. That we will call it as a exotoxin. Okay, so it will release the chemicals, right? Any protein or any chemicals it release, that we'll call it the exotoxin. Exotoxin may be released by two types of the bacteria. One is gram positive, or we can call as gram negative bacteria, exotoxin. Okay, then the special thing is endotoxin. So what happens in the endotoxin is it is going to be released only by the gram negative bacteria. Gram negative bacteria. Why? I will tell you. Okay. See here. So whenever we are talking about the gram positive bacteria or gram negative bacteria, it is having the peptidoglycan layer, right? See here, it is having the thin peptidoglycan layer, right? Which is gram negative bacteria, peptidoglycan, right? So above that peptidoglycan layer, it is having one special coat. Okay, it is having some special coat that we will call it as the glycopolysaccharide, right? Glycopolysaccharide coat, polysaccharide coat, right? So that is what acting as an endotoxin. Okay, so that is acting as an endotoxin. See here, it is inside the part of the bacteria, right? Inside the part of the bacteria. So that is what we will call it as the endotoxin. Means part of the cell wall, right? Yes, cell envelope we will call it as. Okay, cell envelope. Endotoxin is a part of cell envelope. So whenever this bacteria went inside the body, this bacteria is going to be burst. Okay. So when it bursts, the glycopolysaccharide is going to be secreted or removed in the blood, then it is going to cause the disease. So that type we will call it as the endotoxin. Okay. The endotoxin is going to be spread by only from the gram negative bacteria. Okay. So I think you understood this. If you have any question, you can ask me. Do you have any questions here, student? At least tell me in the chat box whether you have any questions here. Okay. So how is your studies going on? Can anyone answer me in the chat box? Okay. So let us come to the common diseases. Okay. So let us come to the common diseases, bacterial diseases. Okay. So see here I have given the easy formula how to remember that. Okay. So mnemonic, Takla, Papad, Dawat, tea. Okay, student, you can remember like this. Okay. So see here, typhoid. Can you tell me where the typhoid is caused by? Salmonella typhi, right? The typhoid is caused by the Salmonella typhi. Okay, student. Then anthrax, then cholera, leprosy, pneumonia, plague, dysentery, diphtheria, whooping cough, tetanus, tuberculosis. Okay, so these all are bacterial disease. In the exam, how they will ask about the disease means they will give you some viral disease and the bacterial disease. Okay, in that they will ask you to select only the bacterial disease. Okay, so that is why you have to remember which are the bacterial diseases. Okay, so remember here typhoid, anthrax, cholera, pneumonia, plague, dysentery, diphtheria, whooping cough, tetanus, tuberculosis. Okay, so these all are caused by the bacteria. Fine. Okay, can anyone tell me the cholera is caused by which organism? Cholera is caused by, can you tell me, name the bacterial name? Cholera is caused by bacterial name. It is Vibrio in shape. Can anyone guess Vibrio in shape? Can anyone answer me? It is Vibrio in shape. Cholera. Yes. So Vibrio cholerae, right, student? It is caused by Vibrio cholerae. Vibrio cholerae. Okay, fine. So let us talk about this. Okay, so fine. Let us talk about the bacterial diseases. Typhoid. Okay, so what you will remember when the typhoid name came? Fever, right? Yes. So it is going to cause the fever. Okay, what is the causative agent, student, here? Salmonella typhi. Okay, so the typhoid is caused by Salmonella typhi. So it is very, very important. The causative agent for the typhoid. Okay, so typhoid and how it is going to be transferred. So student, what are the things we are studying is the disease, then how it is going to be transferred, and what are the symptoms of that. And in some disease, we are going to talk about the diagnosis test also. Fine. Okay. So it is going to be transferred to the fecal and oral matter. Okay, it is going to be transferred through fecal and oral matter, this bacteria. So that is the Salmonella typhi. So then which are the symptoms here? So constipation and abdominal pain and sustained fever for 39 to 40 degrees Celsius. It is very, very important. Okay. And intestinal perforation and loss of the appetite. Okay, so these all are symptoms of the typhoid. Okay, that person is the person who is having the typhoid fever is having 39 to 40 degrees sustained fever, continuous fever. Okay, so then coming to the intestinal perforation and constipation and abdominal pain. Okay, so these all are the symptoms. So then how the diagnosis is going to be done? Okay, here the diagnosis is through the Widal test. Okay, the Widal test is very, very important. Okay. So very, very important. The typhoid, the diagnosis test is a Widal test. Okay, fine. So then let us come to the second bacterial disease. So that is the pneumonia. So student, what the things come to your mind when we call the pneumonia? So starting when we come to the first wave of the coronavirus, the people's having the fever also, and again they are having the pneumonia also, right? Yes. So see here, but it is a bacterial disease we are talking about. It is going to be caused through the Streptococcus pneumonia. Pneumonia is related to the lungs, right? Yes. So how it is going to be transferred? So it is going to be transferred to the droplet infection. Okay. So droplet infection usually through the sputum of the patient. Okay. Sputum. 1 minute. Sputum. Okay. My God, 1 minute. So if the person is having a pneumonia, okay, if he spit anywhere, okay, if he spit anywhere, through that it is going to be caused the disease. Okay, so that is what we will call it. It is a droplet infection. It is going to be caused through the sputum. Sputum. Okay, if we spit anywhere, through that sputum, it is going to cause the disease. Okay, so which are the symptoms here? See here, chills. Okay, chills. In the night times, you will get the chills and sharp chest pain and shortness of the breathing, obviously, right? So it is going to affect the alveolar of the lungs. It is going to cause the shortness of the breath. Okay, then fever, and coming to this is a very important thing. So that is the bluish and grayish lip and nail. Okay, the person who is having the this pneumonia, the person's lips and nails will get turned into the blue color. Can anyone tell me what is the reason of turning into the blue color? Obviously, there is no exchange of the gases is going to be takes place, no, right? So because the no oxygen, there is a no exchange of the gases is taking place, the carbon dioxide concentration is going to be increased where there is no oxygen concentration, right? So because of that, it is going to turn that into the bluish and grayish lip and nail color. Okay, so then how we are going to be diagnosis process? Which are the diagnosis process for this? See here, chest X-ray and the CBC. Okay, chest X-ray and the CBC is going to do for the pneumonia. Usually they will do the chest X-ray. Okay, so how much the lungs was damaged, they are going to get that. Okay, so I think whenever they do the chest X-ray, they will give the scores, right? Based on this scores, they will get to know that how much the lungs was damaged. Okay, fine. So this is about the typhoid and the pneumonia that is we have learned in the bacterial disease. Okay, so do you have any questions here in the bacterial diseases? Do you have any questions in the bacterial diseases, student? Can anyone tell me? Sandhya, how are you, Sandhya? Jainvi Kudra and Sonalish. Do you have any questions in the bacterial diseases? In the bacterial diseases, we have studied about the typhoid and the pneumonia. Pneumonia is related to the lungs. Okay. So alveolar region is going to be damaged here. Okay. So in the alveolar region, the pus cells is going to be, for the fluid is going to be filled here in the alveolar region. So those that's why the exchange of the gases is not possible in the alveolar region. Okay. The fluid is going to be accumulated in the alveolar. Okay. Fine. Then we'll go to the viral diseases. Okay. So which are the viral diseases? Can anyone tell me which are the viral diseases? You know. Just think. Can anyone tell me viral diseases? Which are the viral diseases, students? I need answer from everyone. Deepti Maye, Jainvi Kudra Jain, Sandhya, Sonalish, Subbalakshmi, Anshika Tiwari. Just try, can anyone tell me the viral diseases? Viral diseases is the name. Viral disease names. Fine. So see here, smallpox is a viral diseases and chickenpox, common cold, measles, mumps, hepatitis B that is related to the liver, and polio, you all know that polio, and rabies, now only we studied dengue fever, it is transmitted to the vector, right? Transmitted through vector. And coming to the chikungunya and AIDS, right? These all are viral diseases. Okay, student? Yes. So you have to remember this viral disease. I have told you before only, right? So in the exam, they ask the questions like 2 bacterial diseases they will give you and 2 viral diseases they will give you. They will tell you to select the option which is containing only bacterial disease or only the viral diseases. Okay, that's why you have to remember all the viral diseases and the bacterial diseases. Fine. So in the viral disease, they are given the common cold. Okay. So common cold, all of you know, all of you have experienced the common cold, right? So you know what are the symptoms of the common cold also, right? But we don't know which is the causative agent, right? Yes. See here, the causative agent is rhinovirus. Okay, causative agent is rhinovirus. We know that how it is going to be transmitted. If my friend is having the cold today, means I will get the cold after 2 days, right? Yes, it is going to be caused through the droplet infection, right? Yes, it is going to be caused through the droplet infection by sneezing or by coughing through the droplet infection, right? Then what are the symptoms? We all know that what are the symptoms, right? Yes, running nose, we'll get the running nose, and red eye, and we'll get the fever. Obviously, we'll get the headache, right? And cough and the sore throat, right? In that when we are having the cold, we are not able to talk also, right? Some persons will get the sore throat, right? So these all are the symptoms of the common cold. So here the important thing is, you know the symptoms, but you have to remember the causative agent, rhinovirus. Okay, fine. Then coming to the protozoan disease. Okay, so protozoan disease. Can anyone tell me which are the protozoan diseases? Can anyone tell me which are the protozoan diseases? How many of you know that? Can just think and remember. I need answer from you all. Protozoan diseases. Protozoan diseases. Now only we have studied, now only I told you that African sleeping sickness or American sleeping sickness. So that is by the trypanosoma, right? Trypanosoma is the nothing but protozoa. Okay. So we are going to talk about the protozoal disease, protozoan disease. Okay. So malaria. Okay. So everyone thought that malaria is going to cause by the from foul smelling air. Okay. Foul smelling air. So that's why they are given the name as mal air. Okay, mal air means foul smelling air. Okay, they thought that the malaria is going to be caused through the foul smelling air. Okay, but no. Okay, so one scientist, so that is called as a Leveran. Okay, Leveran scientist disproved that it is not going to be caused by it is not going to be caused by the foul smelling. It is going to be caused by the protozoan. Okay, so Leveran. So another scientist, Ronald Ross. Okay, Ronald Ross is a scientist, is an Indian scientist. Okay, he's proved that it is going to be transmitted through the female Anopheles mosquito. Female Anopheles mosquito. Okay, student, the malaria is going to be caused by the female Anopheles mosquito. Why only the female Anopheles mosquito? Why not the other mosquito? Why not the male mosquito? We know that usually the mosquito will feed on the plants, right? But here what will happen? During the reproductive stage, this require the proteins. Okay, this require the protein, the albumin protein. On the time, it will feed on the blood. Okay, so that is the reason for the female mosquito will feed on the blood. Okay, it requires the albumin. That is the reason. Okay, so it is going to be caused by the plasmodium. So students, in the India also we have got the malaria, but in the India, the malaria is mainly through the two things. One is the falciparum, another through the vivax. Okay. So here you can see the falciparum. Okay. Here the malaria is going to be caused by the three species. One is the Plasmodium falciparum, Plasmodium malariae, and Plasmodium vivax. Okay. These are three are the causative agents of the malaria. But here you can see, Plasmodium falciparum is a malignant. Okay. It is a malignant. It is very dreadful. Okay, malignant. So in comparison to the malaria, sorry, Plasmodium malaria and the Plasmodium vivax. Okay. And coming to the malaria, you have to remember here, this is the only species which will cause the malaria to the primates. Okay, to the primates. All the primates it can cause the disease. Okay. So this is about the malaria. So in the next class, we are going to talk about the life cycle of the Plasmodium and all the things in the next class. Okay, student. So this is for today. Do you have any questions? You can ask me. Otherwise, we will end the class. Okay. We'll end the class. So you can give me your valuable feedback. If you have any questions, you can ask me. Okay, students. Yeah. Thank you so much. In the tomorrow's class, we will talk about the other diseases, protozoan, platyhelminthes, and the other diseases, and we'll go to the immunity. Okay. So, do you have any questions? Means you can ask me, or else you can leave the class. Thank you so much.